Good afternoon, and appreciate the opportunity to tell you about our exciting technology. So, Synpromics, we're the leaders in gene control. If you don't believe that yet, hopefully by the end of my talk I'll have convinced you. So our whole focus is on providing precision control over genes when they're delivered to the body in vivo as part of human health and therapeutics. And we do this to enable safer and more effective cell and gene therapies through our proprietary genomic and bioinformatic and intelligent data-driven design systems. And I'll describe those in overview and show you some data. But we've created a totally revolutionary approach to providing precision in gene control. It enables a new level of control, stability, and precision of genes, much greater than the viral promoters like CMV and others that have been used now for decades in the industry, providing greater efficacy and potency and also improving safety profiles. And this is all powered by a technology engine that we've developed that's called PROMPT, and this is a multi-dimensional bioinformatic toolbox that incorporates our intelligent data-driven design, our capability for building promoters, for screening promoters and validating those promoters. We're able to work with any organism, any gene, any tissue, and under any condition, and design promoters that are active under each of those specific conditions. We're also working in the bioprocessing space to use our technology to improve the yield of protein production, but also to develop stable producer cell lines to support gene therapy, and I'll talk about that shortly. And obviously, to be able to do all of this work, it demands a highly effective multidisciplinary team of incredibly intelligent scientists who know much more about these things than I do and put me to shame and impress me every day with the data they generate. We're a partnering model business, so we create these novel technologies, we patent our technology, and then we license it out. So we've now got an increasing number of partners that have come on board. We're working in over 20 commercial programs across gene therapy, cell therapy, and bioprocessing, and these are just some of those names that we can disclose, and obviously one or two boxes that I'll leave you to have to try and guess what names might be underneath of those. So in terms of the company and our vision, if you listened to the panel yesterday, I tried to explain a bit about how we see the world and how we see things going. And very much at the moment, we're on the far left-hand side here, where we're creating novel constitutive promoters that focus on design criteria around specificity, strength, and size to tackle more increased precision in monogenetic diseases. But for us, that's generation one. That's only the start. What we're now starting to create is inducible on-off control promoters, the way you can toggle expression and have expression that's dose-dependent based on the concentration of the inducer. And we're now moving on to the third generation, where we're starting to look at intelligent design for greater gene control, to design logic gates and to start to think about more complex multi-gene cassettes, with the ultimate aim then of being able to manipulate and engineer whole genetic pathways and circuitry to go into the body to treat more complex diseases. So as I said, it's all based around this system that we've built called PROMPT. This is terabytes of data that encapsulates everything that's known about gene regulation in the eukaryotic system, that enables us to mine this information using our machine learning and AI techniques to be able to do this, so that when somebody comes to us with a specification of promoter, we can go into this analysis cycle, create and design whole libraries of many millions of candidate promoters, barcode those promoters so that we can screen them either in vitro or in vivo, and generate a huge body of data that then goes back to populate our PROMPT design engine. And through an iterative process, the outcome is a collection of highly specific, custom-designed synthetic promoters which are patentable and perform according to the design brief. And I'll show you now some of the data, but firstly, there are many outstanding issues that we're still faced within gene therapy around variations in gene expression and efficacy that we can address. Questions around persistence of expression over a patient life. Is it one dose and done, or are we having to repeat dose? So we believe we can address that through the stability of our promoters designed for long-term expression. Is having options to create and control genes to the expression strength that's relevant to the disease. So it's not almost always the strongest expressor you want or the most protein, but it may be a physiological level. And so to be able to titrate out using our promoters to the right level, we give that level of control, as well as targeting specifically to the cell type of choice or avoiding expression in detrimental targets, and then the ability to induce and regulate through small molecules. So just three quick examples here in muscle RPE and liver, where you can see in comparison to control promoters in red, then we're able to get exceedingly high expression levels compared in muscle compared to CMV and CBA, with promoters that are much smaller in terms of size, so promoters in the 200 to 800 base pair size. Same in the RPE, where we've got small, specific, highly active promoters. And then in liver, we've got promoters around the 200 to 300 base pairs, where we're seeing levels in vitro anything up to about 75 times higher than the LP1 promoter that's currently in the clinic in hemophilia studies. And some data we're now able to share is some of the in vivo data that we've generated with one of our partners, taking some of our liver-specific promoters into a mouse model and showing that the in vitro data translates into an in vivo model, where we've looked at expression from an AAV vector system, expression of SEEP over a 2, 4, and a 6-week time period, and showing that we're getting a dynamic range of expression from a range of different promoter candidates, and are able to show expression levels anything up to 45 times greater than promoters that are currently in use in clinical trials. And the far right-hand graph shows that this is not associated with a change in DNA copy number. So the copy number per cell is essentially equivalent across each of these promoter candidates. So highly selective, highly powerful, in vivo validated synthetic promoters that are now being developed in collaboration with our partners. The other area I mentioned is that our ambition is to develop a stable producer cell line to overcome the inefficiencies that we currently face with being able to scale up AAV lentiviral retro manufacture. To enable to do that, we've had to develop a whole suite of tools that will enable us to incorporate those into stable cell lines. And so I'm just going to show you examples of the tools we've built as relevant to bioprocessing, but also some of these could be repurposed for therapeutic purposes. So the first thing, obviously, if you have a multi-gene cassette, not all genes need to be expressed at the same level. Some need to be at basal level, some need to be incredibly strong. So having promoter options in a dynamic range is hugely important to be able to orchestrate and get the stoichiometry of expression appropriate. So you can see here that in a 293 system, we've got a dynamic range of over 50-fold in our promoter candidates, some very weak basal level expression up to super strong expression, and you can see CMV there in the middle as a comparator. So we're able to get extremely strong in comparison to CMV. The next stage is that some of the genes associated with viral vector manufacture are toxic, so we need to put those under tight control and inducibility. And so we've now created a whole library of inducible promoters that are induced by a variety of different elements. These are either environmental, like hypoxia, or biological, or chemical. And each of these promoters is around 200 base pairs, so they're not huge in size, they're incredibly small and tight. And they show an expression profile against CMV of around 6 times greater. And there you can see the induction response over a 24-hour period of those promoters in comparison to CMV in orange. And so these big key issues has been to create promoters that when they're off are silent, and when they're on, the amplitude of expression is significant. And we've been able to achieve that and can show that here. Even more exciting is to say, well, what about being able to now build logic gates and design algorithms? So here's some examples of promoters we have created, which are actually AND gate logic. So it requires inducer A and B in order to fire that induce that particular promoter. And you can see here that in the uninduced state, the promoters are silent when only one of the inducers is present. there's minimal expression, but when both inducers are present, then the gene is activated, the promoter is activated. And it also shows here that activation is dose-dependent. So the stronger the concentration of the inducer, the higher the regulation of the gene. So we can now think about modulating gene expression based on concentration of the inducer itself. And just one more example of that here, which shows that when the promoter is uninduced, it truly is silent, and when the promoter is turned on, then it's significantly turned on in comparison to CMV. And if I'd been able to quickly recall my presentation and add in another slide, I got a slide from the guys in the lab this morning. So my challenge to them had been, well, this data in theory with GFP, it looks great. The ultimate test of this is to express a toxic gene under the presence of an inducible promoter and show that in the absence of the inducer with a toxic gene in the cell would still grow and the viability would be as high as control. Add the inducer and then let's see what happens to the viability of the cell. And they actually have emailed me a graph this morning that actually shows that in the absence of the inducer with the toxic gene in the cell, we get really high viability. There's absolutely no leakage, no cell death. And then when the inducer is applied, then viability drops, there's cell death. So we know we've positive confirmation that these are incredibly tight and controllable systems, which is great from my perspective. So as I said, our model is partnering. So we do establish a number of partnering deals with companies that gives our partners access to this technology that we're developing. It enables us to work with them, either to provide them with promoters that we've already developed and give access to evaluate those promoters, or we'll custom build to a particular design brief promoters that are required for a particular indication or product. Because these promoters are totally artificial and don't exist in nature, the IP position around them is incredibly strong, which obviously helps to secure the IP position around any product that utilizes those promoters. So from a commercialization and IP protection position, it adds significantly to our partners' patent portfolio. And we believe it also gives our partners the best chance of designing a product that's going to have best chance of success in the market, rather than living with the inefficiencies and compromises that we've been taking with CMV and CBA and other promoters to date. And obviously it's access to a world-leading team, expert in bioinformatics and machine learning and genomics to actually apply that knowledge to help in the design and development of your product. And that's been proven now by, as I say, the 20+ commercial collaborations that we've got in place, and the fact that many of those companies that we're collaborating with have come back for multiple programs with us, not just the ones. So in summary, we've developed a revolutionary technology to provide a precision in gene control that's unprecedented in comparison with what's been available to date. We've got a team of exceptional scientists and apply their scientific rigor to come up with great novelty, and I think through the logic gate promoters we're developing and the inducible systems we're developing, I think that's self-evident. And we provide this technology and access to that technology to our partners to hopefully advance gene and cell therapy into much more complex diseases and much more complex genetic challenges than is currently possible today. We've recently moved into new facilities on the outskirts of Edinburgh. So this is a picture of our brand new labs that we've taken over one of the floors in this building to expand the team and take the business forward. So happy to take any questions.